Welcome back. In this video, I will be going over the Watson and Crick model of DNA and some stabilizing forces. Let's zoom in on our double helix. Firstly, we have our pyrimidine, thymine, and cytosine. They have just one ring. Next, we have our purines, adenine, and guanine. You can see that they have two rings. Thymine and adenine are base pairs, and I've highlighted that they are connected between two hydrogen bonds. Cytosine and guanine are base pairs and are connected by three hydrogen bonds. Now, how about those hydrogen bonds? Well, these bonds are too weak to hold the molecule together, so what they actually do is provide specificity when it comes to the actual pairing. Let's move on to other structural components, the phosphate backbone and the sugar component. The ribose/deoxyribose sugar is made of 5 carbons, 4 of which have significance. 1' connects to the base, 2' distinguishes between deoxyribose and ribose, 3' has an OH that bonds to the phosphate, and the 5' carbon bonds with the phosphate oxygen. This phosphodiester chain of bonds forms a sturdy structure that helps hold the strands together. Now, I mentioned the 5' and 3' ends bond to the phosphates. You can see from my little drawing how they connect and are directional. So here I have drawn a simple modular structure of DNA. Remember what I said about the 5' and 3' having a direction? They move towards the 3' end. This is where new bases are added. That plays into the concept of being antiparallel. This means that the strands are basically going in opposite directions. Now, I'm going to be talking about another stabilizing feature of DNA, base stacking and how it works with hydrophobia. So think about the bases as flat steps on a ladder which are perpendicular to the phosphate backbone, which represents the frame. This overlapping of the flat base pairs creates an interesting interaction with water as the bases push said water away. This base stacking maximizes the hydrophobic interactions of the bases, which greatly stabilizes the molecule by keeping it in place. Next, I want to quickly touch on the components of the secondary structure of DNA. The major grooves are much more easily accessed by proteins, while the minor grooves are just the opposite. Their smaller size makes protein interactions more difficult in most cases. Next is the handedness of DNA. Wolfie is demonstrating with her arms the direction that the DNA is going. This is to the right, so it's right-handed. This one's going to the left, so it's left-handed. Whew! With all that model stuff out of the way, let's talk about the influence of sodium on the phosphate backbone and Tm. So the negative charges on the phosphates generate a lot of repulsion and destabilization, so when the sodium comes in and neutralizes them, it acts as a sort of shield to the phosphate's negative charge and keeps the sugar-phosphate interaction minimal. So normally there's a lot of steric interactions going on, and it's really destabilizing. So with the addition of sodium, the molecule gains stability, so then therefore the Tm raises due to the increase in said stability. And so it will take a lot more heat to actually denature DNA that is in a sufficient sodium buffer solution as compared to DNA that is in a sodium deficient solution. As you can see by these charts here, the Tm will shift over to the right more. So thank you. That is my presentation on the Watson and Crick DNA model and some stabilizing forces. Thank you.